We're to the last part of this chapter. So we're still continuing with organic compounds. So we went over the carbohydrates and lipids. Now, the third one, the third macromolecule or the nutrient is proteins. This is slightly different from the other two by having nitrogen in it. So proteins also have carbon, hydrogen, oxygen, like carbohydrates and lipids, and it has an extra nitrogen. There's many different functions in the body. Okay, so and that's the reason we have to take proteins in our diet. The reason is not for energy. Carbohydrates, especially glucose, is broken down to release energy. But there are many essential, important functions that proteins have in the body. And their function depends on the structure, on the 3D shape of the protein itself. Protein is a very polar molecule. It has many side units sticking out, which contain positive or negative charges, and that interacts with other molecules. So it is highly interactive. Normally, proteins are huge, they have globular structure, and the function depends on their outermost covering, I should say, for lack of a better word. So if the protein were to be unraveled, okay, that looks something like this. So it is like a chain or a pipe of some sort, like a ribbon, and then that folds up in a specific order to make a protein like this. So, for any reason, if the protein is unraveled, okay, then it no longer has the capacity to carry out the function that it is normally supposed to. That is called as denaturation. So the unraveling or unbinding of the protein is called as denaturation. And denaturation can occur inside the body due to several reasons, like increase in temperature or increase in salinity, like salt content, or if the pH is changed. So that's a very important factor for protein function. Um, Earlier we discussed that the pH of blood has to be maintained between 7.35 and 7.45. And then we'll talk about different systems like digestive system, where the food is broken down because of proteins called as enzymes. And those enzymes are very specific in the organ that they are supposed to work. For example, if you have food in the stomach, there are enzymes which can break down food only in pH 2, which is highly acidic. Whereas once the food goes to the small intestine, the pH changes to about 7.8, which is extremely different than what it is in the stomach. So we'll talk about that, how protein structure depends heavily on these three factors, structure and function. And pH is one of the most important factors in that. So the basic structure, or the monosaccharides we saw for carbohydrates, the monomers for proteins are called as amino acids. There are 20 different amino acids, and these are arranged in different uh, um, proportions, okay, or they're in different orders to make the different kinds of proteins that we find. So it's a combination of these 20 amino acids. So amino acids sorry, let me go back. So they can be dipeptide, meaning two amino acids coming together, another amino acid forming tripeptide. Then you have a chain of peptides, amino acids called as polypeptide, and that forms um, a secondary structure, and then it folds upon itself, forms tertiary structure, and then we have quaternary structure, which is probably the protein. So these are complicated structures. Similarly, if a protein has to break down, they have to go through the same steps in order to release the amino acids. So they have to unravel from tertiary structure to tertiary structure, quaternary structure to tertiary structure. to secondary structure, and then a polypeptide, and then it breaks down into amino acids. So that is how digestion of proteins take place, and that's why proteins take a long time to digest. Okay, moving forward, one of the most important functions of proteins is uh, enzyme, and enzymes are biological. Enzymes are biological catalysts which increase the rate of chemical reaction. So they are proteins. Enzymes are very specific. Okay, so they are specific to the type of the reaction that is taking place. So they're very specific to the substrate. Okay, we'll talk about what is substrate. They normally they're named after the substrate that they work on and ending in ase. Okay, for example, an enzyme that can break down sucrose is called as sucrase, lactose, lactase, and so on. So we have peptidase, which breaks down peptides, or kinase, okay, which adds phosphate group, and so on and so forth. So they end in ase. Enzyme um, works on a chemical reaction on the substrate. So things that we start with is called the substrate, and then it breaks down. In this case, it breaks down into products. So this is what kind of chemical reaction is this? If you guessed, this is hydrolysis reaction, right? All right. And then we have active site of enzyme, so which is like the whole protein is not going to function as an enzyme. So it has a specific like a groove into which the substrate comes and fits in, and that is called the active site of the enzyme. Enzymes, so that's about enzymes, proteins and enzymes. Then the fourth organic compound is nucleic acid, and we will talk about DNA and RNA. DNA is called deoxyribonucleic acid. Its function is to contain the genetic code or the code for building proteins. Okay, so that is, and it is found in the nucleus, and we'll talk a little bit more about it in the next chapter. It is made up of repeating units called nucleotides, which is made up of three things: a sugar, which is in this case deoxyribose sugar. It's a five carbon sugar. It has a phosphate group attached to it and a nitrogenous base. There are four different bases, A, T, C, and G, and DNA contains two strands of the nucleotides, where these bases are paired up with each other. And it forms, so you have almost like a ladder-like structure, and then it turns on itself, forming the double helix or double helical structure of DNA. And suppose we have bases in one strand of DNA, like AATCUG. A attaches to T, T attaches to A, and C attaches to G. Now, this one is wrong because we cannot have T and U in the same. So let me just put it as U. So in this case, it is going to attach to UU A G A C. Okay, so these are complementary. These are called as complementary strands. U is a base which is found only in RNA. Okay, and T is found in DNA. So let me replace this with T, and then we can have this. So TTAGAC. Okay, so it is complementary. So let me go back and just add this. So they make base pairs with A combines with T, and C combines with G. That would be easier for you to understand. Okay. So ribonucleic acid, RNA, is another nucleic acid which is single-stranded, and it helps carry the information from DNA to the cytoplasm in order to make proteins. So there are different kinds of RNA, and we will talk about three specific RNAs: mRNA, which is messenger RNA, tRNA, transfer RNA, and rRNA, which is ribosomal RNA. And the function of these is in protein synthesis. So we'll talk about this in the next chapter. And these, again, they're complementary. Instead of having T, they have a U. So let me just write it here. So the bases in this case are A, U, C, and G. Okay. So instead of T, you have U. Okay. So this is how a nucleotide looks like. And there is a specific nucleotide called as ATP. Okay, so this is energy-rich compound. It acts like a currency. So this is the base, right? That's A, adenine. This is attached to a 5-carbon sugar, ribose sugar, and then this is attached to 3 phosphate groups. Nucleic acids, a nucleotide will have only 1 phosphate group. This one has 3 phosphate groups. And you can see this squiggly line. This shows that that phosphate group can be broken or made. So if it detaches, then it forms ADP and phosphate, meaning adenosine diphosphate plus phosphate. So this acts like a currency where if there are any reactions taking place where the bonds are breaking, so energy is released, then ADP and phosphate are going to go get that energy and make this bond. If there is energy required, then ATP goes, which is adenosine triphosphate, meaning 3 phosphate groups, that goes there and breaks into ADP and phosphate, releasing that energy. Okay, so the function is to act as a currency, providing the energy in all chemical reactions. Okay. Now let's go into this. List 2 types of nucleic acids found in the body, RNA and DNA. Create a table of similarities and differences. We can do that in the lab if you want to. And then you can help each other out. You can discuss with each other. So this is how an enzyme acts. So this is the active site of an enzyme. So if you have a sucrose, for example, this will come and sit in that active site, and then it breaks, forms whatever products it has to form. And then this enzyme can be reused again. So enzymes do not participate in the chemical reaction. It's only the substrate and the products. Okay, so this just acts as a catalyst. It speeds up the rate of reaction. And the enzyme can be reused over and over again unless the enzyme is broken down by some other factor like pH or temperature. All right, so this is the end of this chapter.